Hello, everyone, and welcome to our class on the genome annotation in G ranges. So today we're going to focus on getting all the annotations of the human genome in a G ranges object where we can do the find overlaps and count overlaps that we learned with our peak files. We can now compare our peak files to genome features. And we'll do that in the next class. But first, we have to learn how to look at the vast amount of information about the genome embedded in genomic annotations. Where are genes, where are starts and stops, and we'll look at a bunch of it here. You will need the GenCode GTF. There's information explaining how to get to that. It's the GenCode annotations GTF. And so we already have it here for class. So we're going to set the base path and load this in with our first genomic ranges import function, where if you use rtrack layer import, it's going to open this file and read it in as a genomic ranges object. Okay, and I'm going to call it GenCode GR here, defined by importing. This is like read TSV. We're going to import. Actually, we used our rtrack layer import for our peak files previously as well. File path I just have set so we can do this. You would want your file path just this to be where the GTF is on your computer. Okay, so load that in. It takes about 5 minutes, and we're going to dissect it out and learn how to load it faster later. But for now, we got to do it. Okay, so while that's running, we can take a look at what we get here in the GenCode GR. Again, it's a long G ranges, but has a bunch of good information in the element metadata with gene name, gene type, gene ID. And so we'll start by just going in and looking at this stuff and try to find long non-coding RNA annotations and mRNA annotations and what else is in here. Okay. So we talked a little bit in the genomic ranges class about seq names, which is sort of the header of the chromosome. So it's attached to the start and stops, have a chromosome identifier. And we can see here all the chromosomes, X, Y, and M as well for mitochondrial. So that's great. So seq names can tell you just kind of what you're looking at on the chromosome level. And we can table this as well. So we're doing seq names as the first function, and then we're going to table it here and pipe it to head, just to look at the top of it. Okay, so we can see when we do that, we get that information about the chromosome length as well, or sorry, how many features there are for each chromosome entry. So for chromosome 1, there are 265,465 entries, transcript IDs, exon, introns, all that good stuff is in there. Okay, so, and then let's say we wanted to just do the table, the seq names for a specific one. We can index out of here and see what index 25 is the mitochondrial chromosome, and it has 143 annotations in it. So this is really handy and also good ways of looking at your data in overlaps and other ways, but we'll get to that. Okay, so we can also index into this. So we've made this GenCode GR object, and we can go into the @element data and look at the gene ID entries. And we can do the same in our environment by opening it up, and we can see, there's all this different way of looking at it, but we do see a gene name here, so we can get the entries for for gene name or gene ID. We'll do both of them. So here we're just doing, we're indexing @element data, which there is a quick way of doing this where you can then go list data, and then we're doing gene ID here. There's this little button that you can click, and it'll tell you how to index into that down in the console there. So that's what we're doing at element data gene ID. You can also index to it with double square brackets. And let's take a look. Okay, so we can see these are the gene IDs from Ensemble. They have these long numbers starting with ENSG for Ensemble genes. And we see this gene has 24 features, so it could be 24 exons, it could be other features associated with that gene, but it's telling us the number of features for each gene. And then for gene name, same thing, but we get the actual name of the gene, and we'll look at that some more. And then we can also, so we're indexing into at element data and then indexing into each of these features in there. These are good things to start to remember because we're going to use them a lot for the next classes. So okay, so we can index into transcript name, see what we get there, and we get the name of each sort of individual transcript that has probably one gene associated with it, with gene ID. Yeah, that's that. And then so this is, we're kind of doing some index looking into it, and we'll do more of this in a second, but let's just take one feature and turn it into more of a format we're typically used to seeing. And so we can make GenCode info here, and let's find out the gene name information, which is in at element data. And so if we do that and use the pipe to send the information in that column to this as a data frame, as.data frame is a function. And if you leave it blank from the pipe, it knows to just run on what comes out from here. So we can do that. And now when we look at it in our environment here, we can click on it and see each gene name in here. And so you can also filter and look for your favorite gene. Mine's fire, so let's say, and it has 155 entries in it. And you can then go in and look and see what those are in just a minute. Okay, so what we can do, what we did there with just one line, making a data frame out of that information. GenCode is a big object. So what you can do is just grab out the information you want and make your own sort of data frame of things. So we're just going to call this GR length. Here we're going to get the gene ID like we did up above. We're basically just cut and pasting exactly what we did, except for we are indexing into at ranges here. And that's the the start and stops, and we want to get the start and width, which we can infer the stop from. And then the seq names as well. So pretty much main places you'll want to index are at element data to get your type of gene name, transcript, that stuff, and ranges to make sure you get the positions of those features. So each feature has a start and a stop associated with the chromosome, and then you can index in and get those values. So if we run this, we get a bigger data frame here, where we now have 7 variables, and we can see the type of gene it is, and whether it's protein coding, what type of, where it starts, its width, and chromosome. These are the key features that everything is associated with. And so we can start looking at how our peaks overlap with these ranges here and with the ranges in our peak files that we just went over previously. Okay, so let's now take it one step further, and we can use filter to make an object that we might want to use. Let's say we want to overlap our chip peaks with mRNA genes. So we might want to make an object of where the start and stops of mRNA genes are and overlap our peaks with that. So we're doing that here with filter as the function. And this data frame we just made here is what we're using as our data. So that's the first input variable there, is that data frame. And then in there, what we want to look for is something that is called protein coding and has type equal to gene. Okay, there's lots of ways to do this, and we'll do it other ways as well, but this is just a fairly straightforward logical way of saying, go in and give me the, in the gene type column, protein coding, and type gene. So type here, it's only going to grab gene, and then only protein coding. Okay, so if we run that, we get an object called mRNA data frame. And so we will see that all of the gene types here are protein coding. And you can select for ribosomal or other features as you explore all the different annotations in there. Mostly the key thing to focus on is just how to index and make objects out of specific parts of GenCode annotations. Okay, so we can then now make a histogram real quick of what's the distribution length of mRNA genes. And so we can just do that with histogram, and we'll take the log 10 of it from the width column. So that'll tell us how wide the peaks are, or the features are, and we'll have 60 breaks in there. Should be coming out. My window wasn't big enough, so it didn't plot, and it gave me that error. So good to know. So here we're back making our mRNA data frame, and we're going to plot the width of the protein coding genes in that data frame. Okay, and so if we do that, we can see what we get here, that 10,000 to a million here, and the distribution is pretty much Gaussian. And this is just saying how often the gene is that size. So very few are very small, and very few are of this large size, and most of them sit here in the middle. Okay, so we can do the same thing for long non-coding RNAs. Shown here with filter. We go in there, and now instead of protein coding, we're going to look for long non-coding RNA gene in our GenCode GR length. Here we're going in, we're going into gene type equals long non-coding RNA. So here we could filter and look for, no, gene type is here, sorry. And look for all the long non-coding RNA transcripts and just filter to those is what we're doing basically. So that, and then type is gene, because there's exons in there and other things. So we just want to know the gene locus for lncRNAs like we did for mRNAs. Okay, there we go. And now we can plot the histogram of that, and we could see the figure sort of shift to the left. And we can see that long non-coding RNA loci are a little bit smaller, but have sort of the same Gaussian distribution of widths. We can also take the mean of that and just see what the value is. So the mean long non-coding RNA locus is 32.5 kilobases, and the mean mRNA width is 68. So like almost double. So they're shifted by their means like twice as wide. Okay, so then we can look into sort of keep indexing into this element metadata to get comfortable with it. And we're going to look at how many types of genes are there. They're in the at type. A lot of this data is on GenCode's website, but we're going to go over the key ones that you would use frequently. And so in here, we can see in gene type, there was gene, which we just indexed into. We said protein coding and gene, and long non-coding RNA and gene. Those are here in gene. And there's many more transcripts because each gene has multiple transcripts. We have the exons in here. And so that's why we had type equals gene. So we avoided grabbing these other features in there. So much information embedded in there, and I know it feels complicated, but we'll just walk through it and get the objects we need, and we'll go over it again in the next class in overlapping our peaks with these genome features. Okay, so let's go to some more indexing here, where we can go in square brackets here and get actual information out and do a function inside there. So what we can do is with one items is going to say, take the gen code GR. Now remember, this is the first G range. This is a G ranges object. So you can, we did that sort of as a data frame thing. So we're basically going in and grabbing a specific part of it, and you can run a function on it, and then get an object returned to you. So we can go here, we're indexing into GenCode GR, and we're going to run width on how wide or how many entries there are on GenCode GR that are equal to 1. That means the width of the start and the stop of any feature that's equal to 1, which is kind of weird. Maybe a start codon could be there. Yeah, so we're saying do the width function on this, and it has to equal 1. And anything that comes out of GenCode GR that matches that will then be made into a data frame that we can look at real quick. Okay, so let's see, with 1 items. Holy cow, there's 2,369. That's less than the number of genes. So it's not genes that we're seeing here. Okay, stop codon. That could kind of make sense. Maybe where it starts, codons are triple nucleotides, but CDS, like a coding sequence, that seems weird to be 1 nucleotide. I guess this is just to point out no annotation is perfect. There's only like 2,000 of these sort of probably bizarre cases, and some of them probably make sense. We can also see how frequent which ones are. So most of them are from the UTR category. We did the table there on the with 1 items object we just made, and went into type here, and it counted how many entries there were of each type. And so what we can see is most of them are for UTR, but it is weird to think of 33 exons that are 1 letter long and coding sequences that are 1 letter long. Maybe the rest of them we could explain, but you know, nothing's perfect, and it is what it is. And it's good to know how to look there. And we can index into there with a filter too, where we now say, give us in the type column all the exons, and then we can say, here's the weird exons. And now we can look at this, and we have 33 entries of exons that are 1 width wide, and you could go look at them in the genome browser or whatever. Okay, so key thing is indexing, and sort of going back over here and thinking about how these different indexings are working. We can then define a TSS, which is not actually in there. So it doesn't give you an exact start. So there's a start and stop of the gene body. And if you're wanting to look at a promoter region, you have to say how far upstream and downstream you want to go from the start of the gene body there. So there is a handy promoters function in genomic ranges. And let's take a quick look at that. This is super useful, that it'll basically go in. Now we're going to index like we did up here with the square brackets, where we're going to grab specific information, and we made that specific data frame there. To find the transcriptional start site of genes, we're going to run promoters, and it has this extra parameter upstream and downstream from the start. And so if we say 0, then that's the actual start of the gene there. Okay, so same indexing into GenCode GR. We want the gene type, the type category in at element data, basically at this point, is gene. Okay, and that after that indexing is the next parameter. Upstream is 0 and downstream is 0, and then we're going to make that as a data frame. So just a little expansion of what we did above. And we can see that there's 60,609 entries, and that's exactly what we got for the number of genes when we indexed up above. So that's a good sign. We can take a look at it here, and we can see the gene ID, the gene type. So the start of a microRNA you can now sort the starts of different types of genes as well. Long non-coding RNAs and mRNAs are what we'll focus on, but just to show you there's a lot more in there in that gene type category. Gene name. So yeah, we just subsetted the entire genomic ranges, made it into a data frame there by just doing some indexing and running promoters. So that was pretty quick and over a lot of data. So we can do a summary here on the same indexing. So we did this kind of up above, but we're going into GenCode GR and saying how many entries are there, or summarize the entries in type equals gene. So it'll give us what we've got. And we see that it's a genomic ranges of 60,000, like we knew from our environment. Okay, so we got 60,000 TSSs from these 60,000 genes by running this up in promoters. Okay, so now what we can do is make a data frame to find the shortest gene. So we can go here and say, we're going to use tidyverse and pipe in our GenCode GR, like all the annotations in our genomic ranges. We're going to make that a data frame, and then we're going to filter by type gene. We're going to call that GenCode DF. So run that. And we can see that GenCode DF is filtered on the 60,000 genes. And if we look at it, we get the start, stop, and is basically another way of doing that indexing we did up above, but using filter instead of the indexing, and using tidyverse to pipe the data frame forward before in these processes. Okay, so we can also see what the smallest gene is by arranging by the width. So in this GenCode DF we have here, we have the width of each gene. So we could sort here, which is not as fun, so giving it away. But also just how to index to that. So we can use arrange, which will order them by width, and then we can use head. And so if we arrange them, it'll give us the shortest gene first and go to the biggest gene. And so we could use tail and see the biggest gene. So we're using GenCode DF piped into this arrange function and taking that data frame and doing it, arranging it by the width column, and then sending it to head to see in the console. Okay. And so what we can see if we scroll to the top here is there is the smallest gene is chromosome 14, that is only 8 long, and it's a TRD gene. And so yeah, that's just how you could do that. And obviously, as we saw, we could have also done that here in that data frame. But it's good to know how to do this, because you might want to have saved this as an object. Notice we didn't use an assignment operator there. So you might want to assign that to something in the future to save it. Okay, now for the longest gene, this is just doing it a little bit different, where we indexed by width. Now we know what width is because we're piping the data frame in. So now it knows that there's a column named width in there. And we're going to do minus width, which will make the biggest one on top and the littlest one on the bottom. Okay, so same idea. Scroll up to the top of that output. Again, I'm not saving it to an object, but you might want to in the future. And then maybe even filter by a size number greater than, etc. So if we look here, we get RBFFox1, and it has a width of, whoa, okay, so 2.4 million bases. That's a huge gene. So you could now take this and go into the UCSC genome browser, which we'll cover later, and paste it in and see, what is this gene? It's so big, and or sort them by other features. Okay, that's great. So we're seeing that there's a lot to look at here in the human genome. And if we wanted to do this a different way to find the longest gene, we could take our GenCode DF, which has the width of all genes, and use filter with and then use this command max. So it's going to find the top one, and you could do min to find the smallest one. So we're basically doing the same thing we did before. Pipe in this data frame so it knows the column name. That's why we can just say width, and width equals max width in that column. And so it's just the max value in that column. And we have longest gene here, and we can now see the information for it in a different way here. And filter, we could filter on different lengths or ranges in here, which we'll get to. Okay, so we are moving along, and so now we want to get into finding overlaps and or asking other questions. So one way to explore the genome is to simply say, what percent of the genome is exons? So exons are the things that make up the mRNA and or lncRNA and are the key elements. So what percentage of the entire 3 billion letters are they? Well, with the skills we just learned, we can do this with just learning one more, which is reduce, and we can answer that question. Okay, so we have here, we're going to say total exon length in the genome, and we're doing this square bracket indexing again. Again, it's super common. We're going to do this a lot. And we're just going to subsect out this little bit of information we want here. So we're going to go in gen code gr and go into the type equals exon. Before we grab gene and protein coding and long non-coding RNA and gene type. And now we're grabbing exon. I think we can just see it here. If we run that, that these are the different types of, we probably want to run a table on this, but in here we can see the entry of exon down here. Okay. Also, it's a handy thing to note that you can highlight just a small piece of code and run it. It's often good to do that when you have nested things to see, what am I looking at in here? And this is what you're looking at. And we could do a table here and then get the number of entries of each unique one and how many exons like we did up above. Okay, so now the problem is there can be multiple exons in the same position for a given gene. So what we would do as one way of doing it seems like the most straightforward way to me is take the overlap of all the exons in a given position and take the longest intersection. So if they overlap, take the sort of longest width of that overlap, and that's what reduce will do. So we can use the genomic ranges reduce function here, and it'll take anything that's overlapping and take the outer edges of where that overlap occurs. So you could have a small one here, a medium one, and a long one, and it'll take the sort of the longest piece of information that overlapped. And so what we're basically saying is we're going to take the estimate of the genome of the longest exon version in a given exon position. Okay, so we are doing the indexing in which to pipe exon into, so this will have all the start, stops, widths of exon and put it into this reduce function, which will then find any overlaps and merge them with the longest one. And then we will take the width of that output and then take the sum. So that gives us the total coverage of the genome that is exonic in nature. Okay, so now we have this object total exon length here. It's just a value. I don't know how many, what that number is. I can't do the count it that fast. So what we can do is just divide it by the total length of the genome is 3.2 billion base pairs or nucleotides, and divide it by that, and we can find the percentage. And what we find is about 5% of the genome is exons. Cool. That's fun to know. Good dinner conversation, I guess. And so, and if we wanted to plot this, we could just run the same code here, indexing, grabbing just the exons and reducing them. And we can see what the average sort of width of different exons are. And we can do that with a histogram. do width of the exon length, so it'll give us the summary of how frequent a size of an exon is, just like we did with the size of an mRNA gene. Here, I'm just putting some breaks in an XLIM for ease, and we can see that most exons are less than 2,000 long, and some are quite large. Okay. And so if we wanted to find out what's the average size, like we did above, we see that most of them are under 2 kb, but what's the average is we can do mean of the width of exon lengths and put that in there. Because we made this exon lengths object up here, which is just this number that we just had in the values up there. Okay, so we can then do this and see that the average exon length here is 444 letters long. Okay, learning a lot about the genome. It's just really an exercise in indexing and what you can do and what kind of simple questions you might want to ask, and then find overlaps with our ChIP data in the next class. Okay, what percentage of the human genome is comprised by gene bodies? Well, we're really going to just follow the exercise one more time to get this indexing down. Same deal, GenCode GR, subsetting out with the square brackets into the type category of gene. We're going to reduce the gene bodies the same way we did the exons. We're going to take the biggest one that might be in there, because multiple genes might have different gene body sizes for different isoforms. And it's just basically going to take the merge of that, get the width of that range, and then sum it as we did before, and get a number that we can then divide by the total size of the genome and see what's going on there. Okay, so then to find out that, we see that, whoa, cool, 59% almost of the genome is gene bodies. So that's 10 times more than exons. So what we can kind of conclude is that in a gene body, exons are 1/10 the size of the introns. If we take the simple assumption that a gene body is comprised of the exon and introns combined, we now know the length of the exons, and is on average is 10 times smaller than an intron. Okay, so we can plot that. It's really the same thing, so I'll skip over it for brevity, but you can go over it. Really, the key thing is thinking about this indexing and what type of features you want to grab. We're pretty much going to go moving forward, grabbing long non-coding RNA and mRNA gene bodies, and finding overlaps with the ChIP data. We'll do the same thing with the RNA-seq classes. And really just getting the idea that you can take any two things and overlap them now with genome features and peak overlaps. You can find your consensus peaks that overlap in your ChIP data, and then find what features they overlap in the genome by making objects in genomic ranges to index, and we'll find overlaps too. All right, thank you very much, and I'll see you next time, and be well.